Okay. So let's start with the Chloroflexi, or green non-sulfur bacteria. And the reason for that non-sulfur we'll get to in a little bit. By far and away, the best studied member of this group is Chloroflexus itself. And I showed you pictures of Chloroflexus mats the other day, these brilliant orange mats in Yellowstone, which are a combination of Chloroflexus and Roseaflexus. This shows every genus in the group. This is not a broad phylogenetic group. They fall basically into two phenotypic types. All of them are filamentous. Usually you can't see the individual cells within the filament, but they are set up as individual cells. That is, they have septa in the filaments. But you usually can't see them. They're quite flexible and quite thin filaments that are either photosynthetic, or at least phototrophic, or are heterotrophic. And many of the heterotrophs are also thermophilic. The phototrophs, like Chloroflexus, carry out cyclic photophosphorylation. They have a single reaction center complex, a single photosynthetic system. That photosynthetic system is homologous to photosystem 2 in chloroplasts. You're familiar with that from before, I'm sure. Because they carry out cyclic photophosphorylation, they don't have a Calvin cycle. Many of them can fix carbon. They don't like to, but they can. But they don't use the Calvin cycle for this. They use essentially a highly modified version of the TCA cycle called the hydroxypropionate pathway, which is found otherwise in archaea, but is found in this group as well. And so you reduce CO2 and make glyoxylate that way. Most often, though, these things are growing photoheterotrophically, meaning they get their energy from light and their carbon from organic material in the environment. They usually grow in symbiosis or in mats with other creatures where they can get their carbon. If you grow them aerobically, they'll usually just grow straight heterotrophically, and they'll turn off the synthesis of photopigments, and you can grow them as plain heterotrophs, most of them. They are gram-negative in the sense that they have an outer membrane. Many of them have internal membranous sacs, essentially like a single plate of a thylakoid on the inside of the membrane. These things are called chlorosomes. And again, the chlorosomes contain antenna complex, but not reaction centers. And so antenna complex is a protein with a photopigment, right, chlorophyll. And then when the light is absorbed, they transfer from one to another, and that energy state makes its way ultimately to the reaction center where photosynthesis can take place. But that doesn't occur, that photosynthesis takes place at the cell membrane, not in the chlorosome. They also have an outer membrane, but there's no LPS in it, so it's not, they're not usually considered gram-negative. The lipopolysaccharide is found predominantly in proteobacteria, and that's one of the ways people often define gram-negative. So here's Chloroflexus. This is one of the photosynthetic ones. Nice, thin, flexible filaments. You see this kind of thing a lot, where you've got the filaments form a knot. I'll show you some better pictures of knots in a minute. If you see a filamentous organism tied into a knot, you almost certainly have a member of this group. That's kind of one of the diagnostic things you see right away. Commonly found in hot springs. I showed you this picture last time. It's easy to grow Chloroflexus. And we call these things Chloroflexus mats. Turns out they're probably not the primary phototroph in this mat system, though. If you take a piece of that mat and grow thermophilic phototrophs in it, you get chloroflexes. But if you use probes, ribosomal probes, which we'll talk about in a few weeks, and look at the actual filaments to see which ones are chloroflexes and which ones aren't, it turns out that most of them are actually rosiflexes. In mats, these things are associated with cyanobacteria, and it's the cyanobacteria that are fixing the carbon. Chloroflexus is not capable of fixing nitrogen, and so they get their nitrogen also from the cyanobacteria. They are, however, capable of acquiring sulfur from sulfate. And so you need sulfur in the cell, right, for a variety of purposes. And these organisms can fix that sulfur from sulfate. This is why they're called the green non-sulfur bacteria, because you don't need a source of reduced sulfur to grow them. And you might think that in these hot springs, reduced sulfur would be plentiful, and it probably is, but they are capable of fixing sulfur from sulfate, and therefore are green non-sulfur bacteria. Here's herpetosiphon. Herpetosiphon is a mesophilic heterotroph. Most of these heterotrophic chloroflexi are best known from wastewater treatment facilities. Now, they're common in the soil, you find them in sediments and that sort of thing. Almost none of them have been grown in culture. But they're really abundant in wastewater treatment processes during the digestion. So when the raw sewage comes in, usually the first thing you do is really mix and aerate it, pump a bunch of oxygen into it or air into it. Yeah?
What do you find as a chemistry?
So these things they're growing heterotrophically. They're using organic carbon. So they're chemoheterotrophs. They're not chemoautotrophs.
Right, but they're non-phototrophic, which means that they're chemistry.
Yes. Yeah. And so they're using organic carbon, and both for energy and for carbon itself. This is a great overhand knot here, by the way. Yeah.
Why they call it non-sulfur? So they fix sulfur from sulfur.
They can fix sulfur, and so any organism. I shouldn't say any organism, but sulfide is the available source of sulfur that organisms can make iron sulfur centers from and all that kind of stuff. So you need sulfur for a variety of purposes, and it's sulfide that's the readily accessible version. It's the fixed version of sulfur, just like ammonia is the fixed version of nitrogen. Most organisms can't use sulfur and sulfate for their metabolic needs for sulfur. And so just like most organisms can't use nitrogen gas for fixed nitrogen. And so if you can take this oxidized form of sulfur and suck it into your metabolic pathways, that's called fixing sulfur. And since these organisms can do that from sulfate, they're called non-sulfur bacteria because they don't, I mean, obviously they need sulfur, elemental sulfur, but they can use it in this unusual but common form. They can use sulfate, and most organisms can't do that. You don't have to feed them elemental sulfur, and you don't have to feed them sulfide or sulfur-containing organic compounds. They can take that straight from sulfate. Does that answer your question? So these filaments of chloroflexi, we'll talk about them later, are really, really important in wastewater treatment. So once you bubble up air through the sludge, or through the stuff, then you let it settle. You stop the mixing, stop the bubbling, and all the sludge settles to the bottom, and more or less clear liquid stays on top that you can pump out. You treat it with ozone or something, UV light, and pump it out into the water system. These filaments are really, really important for both the flocculation of that sludge and for it settle into the bottom. And depending on circumstances, if you don't have them, the sludge won't settle because it won't flocculate. Or if you have too much of them, the filaments trap bubbles of gas and cause the sludge to float instead of sink, and that creates a nightmare for treating the stuff. So wastewater treatment microbiologists know a lot about these filaments, even though almost none of them are cultivatable. Instead, they have these beautiful keys where you look at different filaments in sludge, and they change how the sludge processing is taking place depending on how much of these different kinds of organisms they're seeing. Enterolinea is an interesting organism. It's a member of the same group, but it's a thermophile. And these organisms are found in bizarre environments. So Enterolinea thermophila was isolated from a thermophilic anaerobic digester treating soybean kernels. You wouldn't think of that being thermophilic, right? But there's so much organic material in this stuff that it heats up. And this stuff sits at 55 degrees in the wastewater treatment facility. It's an obligate anaerobe. And it uses protons as the electron acceptor, and the result is it generates hydrogen.